A great pleasure and an honor to have the opportunity to give the first of a number of what hopefully will be fascinating keynote presentations to this audience. I remember well about a decade ago sharing with Professor Amari and several others in the room the opportunity to think ahead, think big, and think for the future, and to have the vision of a International Neuroinformatics Coordinating Facility like we see here, and this conference kicking off the next phase at a very timely period when the field really needs what this community has to offer. So I'm going to make a few general comments about the big picture that I think we all are aware that there's just tremendous diversity in neuroscience relative to many other fields, and that diversity shows up at many scales. You'll hear from Mary Kennedy about the molecular scale, and you'll hear from me in considerable depth about the macroscopic whole brain scale and its various pieces. And there are all these different modalities with which information is acquired. And what we're seeing is that there's increasing need for comparison and analysis across studies and across modalities, and that's where we have opportunities and challenges. Part of the challenge is that we have these staggering amounts of data as well as the sheer diversity impinging upon us, but what reaches our awareness is a tiny fraction that's distilled into publications. And of the rest of the data, there's lots that's potentially useful, but only a tiny amount of that is accessible in databases or through any other means. What we face then is a situation where data sharing, data mining, and data modeling has tremendous opportunities, but really some big-time challenges. And I want to make an important aside that as we face these challenges, we as a community have lost two key leaders in recent months. And I just want to say a word in appreciation and memoriam for Bill Bugg, a splendid neuroinformatician who would have been here and have been a very lively voice except for his untimely death last spring. And even more recently, a colleague of mine, a psychiatrist, Richard Todd, and a neuroimager whose contributions you'll see a little later, died of cancer. And we have these unfortunate losses. On the upside, we have the splendid news of Sten Grillner being the co-recipient of the first Kavli Prize this coming week, and I think that's tremendous for him as well as for the field. So coming into focus on the major theme of cerebral and cerebellar cortex, we know, of course, that the cerebrum and also the cerebellum together form the dominant structures of the mammalian brain, that they are convoluted sheets in many species, and that the challenge of charting the organization of these structures is the realm of what we call cortical cartography. And that leads me to a brief perspective on a different kind of cartography. We've had about a millennium or so of serious cartography of the Earth's surface, which in its early instantiations led to these charming drawings and conceptualizations of what the Earth was like. And we went through another period last century where there was much greater detail and fidelity of representation of the Earth's surface, but that was in the form of books and atlases that had page after page of different kinds of information. Things changed dramatically when the acquisition and the processing of information went digital and brought us into the satellite and now the GPS, global positioning system, age. There were periods where Landsat was spectacular forms of imagery, but now we're in the Google Earth period, and I find it still boggles my mind that I can zoom in on Google Earth and jump from St. Louis and see where I started a couple of days ago and zoom over to the location of this Congress here, all in a matter of a few seconds. So what's the relevance of that analogy? We've had a century, not a millennium, of mapping of the cerebral cortex in any serious way, starting with the classical anatomists, Brodmann and von Economo and others at the beginning of the 20th century. Then we went through a period where very important books were used to describe atlases of the human brain and other brains, such as the famous Taylor and Turneau atlas. Then we moved a little over a decade, maybe 2 decades ago, into the digital domain where structural MRI, functional MRI, and a growing number of incredibly powerful tools have been able to gather information, typically starting in what we call volume representations, and then converting, as you'll see in detail, into surface maps as an important complement to how we represent the surface, and leading us into opportunities for a tremendous amount of data mining that I'll talk about. So over the past decade, my lab has invested a lot of effort in our cortical cartography theme into the development and utilization of atlases of cerebral and cerebellar cortex of human, non-human primate, and rodent brains, trying to bring these tools and infrastructure into play as useful substrates for analysis and visualization for the field. And our emphasis includes analyses of structure, function, disease, which I'll talk about, and development and evolution, which I'll only fleetingly mention, but are also important parts of the story. So in terms of the organization of the remainder of this presentation, I want to spend some time on a fascinating issue and a challenging issue, coping with individual variability, but also understanding something about its nature. Then we'll talk about atlases, surface-based atlases, and the analyses we can do with them in focusing on examples relating to what we call surface-based morphometry. And then we'll make use of these atlases to bring various kinds of experimental data into a common framework and into public access through databases. So the efforts in my lab have been carried out by a small but very powerful and dedicated group of individuals. Donna, John, Ping, and Aaron are the current team. Heather and James contributed much but years ago. And we've developed 2 sets of tools. Carrot is the software application that is our workhorse tool for offline analyses, and SumsDB and WebCarrot are the online tools you'll hear about later. And then we've had many collaborations over the years as well as we explore and make use of these tools. Let's see if I can get this movie going. Maybe, maybe not. These are meant to be movies of the human brain scrolling through structural MRIs, and we had them working a little while ago. Do you know if there's... Did it work before? I'm clicking on it. Ah, there... Well, imagine, if you will, going from the back to the front of the brain and just seeing a representation of the structure of the brain on the left and a segmentation. Let's not worry about it because it's no, let's just a movie that takes this. That's better too. This volume and runs from back to front to show the complexity of the brain, but our ability to capture the shape of the cortical folds using a process that we call segmentation, and to take that segmented volume and wrap a geometric surface around it, which we call a surface reconstruction, that starts out as a fiducial representation of the three-dimensional shape, but then it can be manipulated and inflated or flattened as we choose. And in order to see the smooth surface, it's nice to have this inflation process, but we need to have a representation of the underlying cortical shape. And what we use for that purpose is a measure that we call sulcal depth. So at each point on this original surface, we compute the distance, whether it's near or far, from this external domain that we call the cerebral hull that wraps around the outside external gyri but doesn't dive into the sulci. So over on this sulcal depth map, you see that the darker it is, the deeper it was in the original surface. And this is essentially a cortical brain print that we will make much use of. And this kind of, there are many ways of getting these kinds of reconstructions and other ways of representing cortical shape, but these are the ones that we will key on. So let's move on to this issue of individual variability. And I'd like to bring quickly to your attention that there are 5 distinct and important types of variability. One is just the differences in overall brain size and shape. Your skulls are different size and shape, and so are the brains that are inside them. But we're much more interested in these more intricate differences in cortical folding patterns. If you look at one pair of hemispheres, left and right from above, and then another individual, you see there are tremendous differences in the details of these convolutions. We'll come back to that, but that gets it on the stage. And then we have another type of difference, which is that the cortical areas that make a mosaic representation on this cortical sheet, those cortical areas themselves are highly variable, 2 to 3-fold variability from one individual to the next. So here, for example, is the remarkably well-defined primary visual cortex, area V1 in the Calparin sulcus, and Andrews et al. And many other studies have shown that the variations in surface area go over quite a range from one individual to another. A third type of variability is what we call function versus folding mismatches. So over here from Amunt-Silas et al. Are maps of area V1 in red and neighboring area V2 in green, and you're looking at it in 4 different individuals, and you can see that the location of area V1 relative to the nearby folds is quite variable, and that poses another challenge. Lastly, but certainly not least, is the expectation that differences in the wiring diagram from one individual to the next are hugely important, because that's likely what makes us think differently and see and perceive differently, and whether those are macroscopic differences in the long-range connections or local differences in the intrinsic circuits, the combination of those derived from genetic influences and experience are what makes each of us a unique personality and individual. So fascinating as it is, we also have to cope with it. How do we compensate for the tremendous amount of variability? Well, one standard approach is to register different individuals to an atlas, and the ideal, if we had our druthers, we would align all of the functionally corresponding locations in any one individual to a common target. So everybody's map of visual area 1 would be precisely in register, and ditto for every other functional subdivision of the brain. But that, of course, is not in the least feasible, because we don't even have good maps of all the cortical areas in each individual. And what has emerged as the dominant compromise is to use structural or geographic constraints to deal with the problem. And there are 2 major approaches. One involves the volumes that I would have shown you in the movie and you saw a slice of, to try to align all the slices in each volume using either low-dimensional or high-dimensional algorithms to achieve the alignment. And the other is what we call surface-based registration, and there are 2 major flavors of that. Now, I'm not going to go into the methodological details, but just to say there are many different variations on the theme. It's quite important, the outcome, but I'll just give you a few highlights as we go along the way. So coming back to an example of comparisons between brains, these are 2 individuals scanned at different times and oriented slightly differently in the MRI machine, and having differences in size and orientation. Following the general idea that was pioneered by Taylor and Turneau, one can register these individuals so that the overall size and shape of the brain is about the same. But when you average across many such subjects, you end up with an average MRI volume that's very fuzzy, reflecting the imperfections in the detailed alignment of gyri and sulci. And that is better appreciated if we look at the same problem but from a surface-based perspective. So here are 6 individual brains which have already been aligned so that the volume's alignment has done as best as it can, so that they're overall same size and shape. But now if you look at the intricacies of these folding patterns, it's very difficult to find where the corresponding little wrinkle here is in other individuals. And even if you smooth out the wrinkles to some degree and look at a partially inflated surface, you see that the problem remains quite challenging. And if we take those same 6 surfaces, we can note that if you spend a lot of time on it, as I and others have done, you can find a few sulci, such as the central sulcus, that's pretty consistent in location and extent and shape. Same is true for the Sylvian fissure, but for many other regions like the superior temporal sulcus, you can see that sulcus in every individual, but as you look at the rear end here, you find it's highly variable in whether it forks or not and how it's located. And in still other regions, the folding is almost random. It's like it's been crumpled arbitrarily like a sheet of newspaper. And this fact of life with regard to human individual variability takes on an even broader perspective if we look at the tremendous variability across species in not just the degree of folding from a lissencephalic manatee to an incredibly convoluted dolphin, but the details of the folding pattern are highly species specific. So let's take a couple of additional moments and explore these issues about when and how folding occurs, how are folds related to functional areas, how do we think about the species differences, and whether individual variability in cortical folding is a heritable characteristic or not, and whether abnormalities in cortical folding might be related to diseases and to cortical connectivity. So there are 2 important parts to this story that I'll tell you about. The first is that it's now possible to monitor cortical folding in human infants that are born preterm. And so this is an emerging collaboration we have at WashU with Jeff Neal and Terry Ender and graduate students Jason Hill and Andy Knudsen. And what happens is that all too many mothers give birth prematurely, sometimes as early as 25 or 26 weeks, when you can see the brain is extremely lissencephalic, and we can capture the shape of the brain by scanning with parental permission in the intensive care unit, and then use a variation on our cortical segmentation method to capture the shape and show these surface representations. And you can see how much cortical folding occurs in this last trimester of birth. And this is an ongoing project. I'm just giving you the thumbnail highlights of it, but I'm using it as a lead-in to a mechanistic perspective and an idea about tension-based morphogenesis, which is, you can't see it there, it was 1997. It's more than a decade old, but if you haven't heard the story, it's worth tuning in for this. So the idea is really quite simple, and it's tied to the fact that the cortical folds develop, they emerge right around the time that long-distance cortical-cortical connections are in fact being established in monkeys and very likely in humans. And the idea then is that every neuron, as it sends out an axon and connects with the target structure, is generating a little tiny bit of mechanical tension. Think of it as a neuron pyramidal cell here is like a fishing pole, and this is a fishing line. And when it latches onto a target, it tries to tug gently. And this is a giant competition by this hypothesis that regions that are strongly connected get tugged together, forming a gyrus in between. Regions that are more weakly connected may be trying, but they're losing in the battle, and they allow a sulcus to form in between. It's an unproven but very attractive hypothesis, and one reason it's attractive is because cortical attention-based morphogenesis can account for both consistency and variability, two of the features I've already mentioned. So the idea is that consistent folding occurs in regions that are dominated by large cortical areas and robust pathways, whereas variability occurs in what we can think of as balkanized regions with many small areas of cortex competing and having different size and different connectivity patterns and allowing different outcomes from one individual to the next. And by that hypothesis, then, abnormal connections reflecting altered strength or pattern of connections may contribute to structural abnormalities that I'll be talking about in a few moments. So let's go from this overview of individual variability to this consideration of how to build an atlas that works for registering using a surface-based approach. And our approach in this that we developed a few years ago is that less is more. That is, because there are only a few regions that are highly consistent, like the central sulcus I talked about and the Sylvian fissure, we use only a small number of registration landmarks to bring each individual into a common spatial framework. I'm not going to go through the details of how we do it. I'll just show you the outcome, which is that we register individuals to a common target, and then we generate this somewhat crinkly version of a hemisphere that we call an average fiducial surface. So it's the average of all these contributing individuals, it serves as an excellent substrate onto which to map experimental data, but we use these mildly or highly inflated surfaces for the purposes of visual representation. And the fundamental point is twofold. One is that this registration process respects the topology of the cortical surface, and that gives us greater fidelity in the mapping process. And secondly, it specifies what the algorithm defines as corresponding geographic locations, and we can use that as a tool for analysis. So here's our first foray into that. We can take the sulcal depth maps that I showed you previously and average them from all 12 of the contributing subjects to this PALS, Population Average Landmark and Surface-Based Atlas. And we get an average sulcal depth map, which is, shows high contrast in regions of consistent folding and low contrast, grayed out or fuzzed out in regions of high variability. We can quantify the variability by showing this map of three-dimensional variability. That is, at each point here, the color represents how much the distribution of three-dimensional coordinates of the contributing surfaces, or we can have a map of sulcal depth variability, how much variability is there in the depth value that I showed you how we compute. And so you can see there's regions of high consistency in the blues and grays, and regions of high variability in parietal, temporal, and frontal cortex. We can add to this atlas maps of what we call cortical geography of identified sulci. So here are painstakingly identified maps of identified sulci, the superior temporal sulcus, and many others in an individual subject, and in our population average in this middle panel done by surface-based registration. And for comparison, we did the same process using the classical or conventional volume-based registration. And in a nutshell, we used this to show that the quality of alignment is about twofold better using the surface-based compared to the volume-based approach, validating the point I made before. So with these atlases and analysis methods in hand, let's do something interesting with them. And the first one is to compare the left and right hemispheres. And since we've registered both the left and right hemispheres to a common hybrid target, we can make these comparisons without being biased for one side or the other. So here is an average sulcal depth map for the left hemisphere and for the right hemisphere below. And the one thing that's most striking is that the superior temporal sulcus here is paler on the left and darker on the right. And that's because the superior temporal sulcus is indeed deeper on average on the right. And the outlined regions in green and blue are the regions that are statistically significant. So what we've identified is a collection of significant hemispheric asymmetries, some like the planum temporale here, which were known before, and others which weren't widely appreciated. So that's just the beginning one analysis. Here's another one that's fun. There are two twin pairs here. Now, if genetic factors contribute to the folding patterns and to the individual differences in folding, then for identical twins, there should be some commonality, some similar shape characteristics. So I challenge you to take case W here, we just named it arbitrarily, and say which of these three others is its twin pair. And I won't take a vote, we won't take the time to do it, I'll just say it's a hard problem. And if you try to make it a little easier by, instead of using these fiducial surfaces, the three-dimensional surfaces, you can analyze the sulcal depth maps that I show on the right here and say, well, which cortical brain print is most similar to case W? It's really hard, I can tell you, because I spent many hours on this. And we actually had a competition between 5 investigators who claimed to be talented neuroanatomists, like myself, against a computer. And the computer was assigned the task of just doing a cross-correlation between each sulcal depth value at each node here versus on each other case, and doing pairwise cross-correlations to estimate or quantify the similarity. And what you can see here, I hope, is that the highest cross-correlation, other than the W versus W identity, case W is most strongly correlated with case Y and X with Z, and that in fact turned out to be the twin pairs. Moreover, when we did our competition, not just between a couple of pairs, but between 14 twin pairs analyzed in just this way, the human experts, 5 of us, each predicted, got it right on about 3 pairs. It was humbling, embarrassing, and shaming to me that I did the worst of our 5. I only got 1 right. And the only thing that made me feel good is that the computer cross-correlation analysis got all 14 right, so it won hands down. But more generally, this provides validation that this kind of quantitative surface-based morphometric analysis gives us an inroad into analyzing things such as the heritability of cortical shape. And this was a collaboration with Kelly Botter and Richard Todd, unfortunately deceased, but a wonderful colleague. So moving on to now not normal individuals, but individuals with a disorder known as Williams syndrome. It's a relatively rare disorder, but a striking one, because the individuals with Williams syndrome have some severe deficits, such as visual, spatial, and math abilities. Other capabilities are spared. In other ways, like hypersocial behavior and very strong musical interests, Williams syndrome individuals are actually very engaging and striking. We approach this from the kind of morphometric analysis that I've already touched upon by looking first at the structural MRIs obtained in collaboration with Ursula Bellugi and Alan Rees, and then using our surface-based approach. And when we looked at the MRIs or the individual surfaces, it was hard to see any real differences. But when we applied the kind of analysis you're now starting to get tuned into, we looked at the average focal depth of Williams versus control, and you see there might be some differences. When we looked at the depth difference map, it was very colorful, but we of course had to worry about noise, and that in regions of high variability it might just be spurious. So we did a t-statistic analysis and then looked for significant clusters, and at the end of the day we found a striking pattern in which 33 distinct abnormalities were identified, 16 on the left, 17 on the right, and it was very symmetric between the left and right hemispheres. And the abnormalities, whether it was controls deeper or Williams syndrome deeper, they were distributed from parietal through frontotemporal to ventral frontal cortex. And out of these 33 abnormalities, I'll talk about one in a little more depth, so to speak. If you look at the orbital frontal cortex, so this is looking at the ventral surface of the brain just above the eye, and you can see that in control subjects, the olfactory sulcus, on average, is pretty deep. And in Williams syndrome, it's much shallower. And we confirmed that in individual subjects, but this is the average. And that actually brought a conceptual tie-in to the issue about what might cause folding abnormalities that I showed before. So these are the regions of significant folding abnormalities by statistical analysis. And then we brought into our atlas framework a modern-day map of the relevant cortical areas using an analysis, a study by Dost Unger, Joel Price, and others, where they painstakingly and precisely identified the cortical areas in this region of human cortex, and we mapped that to our atlas and established that the region of this olfactory sulcus where the folding abnormalities occur include several elongated areas, 13B, 13M, and 14R. And the hypothesis is completely unproven, but it's an interesting speculation that maybe what's going on is that normally the olfactory sulcus folds by the tension-based morphogenesis method, reflecting strong connections, local patterns of connections of 13M and 13B that cause a crease to fold between them, and that in Williams syndrome, the shallower olfactory sulcus might either be because these areas, 13B and 13M, are smaller, or they're disrupted in their connections. We don't know the answer, but this is now a testable hypothesis using modern neuroimaging methods. Let's move on quickly to another disease, autism or disorder. It's actually not one, it's a spectrum of them, autism spectrum disorder, a much more prevalent and profoundly serious problem for our society, with so many individuals having some variation of autism. And what we found in a collaboration with David Amaral and Christine Wu Nordahl is that our analysis approach could identify folding abnormalities in autism, not 33, but just one or two, but they're shown here. And what is striking is that the folding abnormalities differ substantially for low-functioning autism, kids with very low IQs, high-functioning autism, and the difference between these is their location in insular cortex and nearby frontal gyral cortex. And then Asperger's syndrome, a much more mild form of autism, has a very different pattern concentrated in the intraparietal sulcus. So fascinating and many things to follow up, but from our neuroinformatics perspective, I want to use this to tie into the importance of bringing this kind of experimental data into the framework of comparisons with reference maps, some that you've seen and more to come, as well as various kinds of functional data. So just within the three studies, the two studies that I mentioned before, and a third study with John Cernansky, where we looked at schizophrenia and found yet another folding abnormality, having all of these data sets in a common atlas framework and a toolkit for visualization analysis allows us to make very careful comparisons of the location of Williams syndrome folding abnormalities, which are shown in red and green outlines, compared to the different forms of autism and schizophrenia folding abnormalities. So this doesn't answer questions right away, but it gives us a basis for saying, are these abnormalities in similar locations or slightly different? And slight differences could be very important. But we also want to compare or relate these kinds of structural abnormalities to the underlying partitioning of the cortex. And I've already shown you an example and a rationale for why to do it in the connection with the Price Lab orbital frontal areas in relation to Williams syndrome. But that's just one partial map of human cortex, and there are many other maps, including the classical Brodmann map, which we have transferred from Brodmann's drawing onto our PALS atlas. We have a rich collection, a very important data set of probabilistic architectonic maps from the Amuts and Zillis group collaboration, and those are presented to the world as probabilistic volume representations, and they have provided those data sets to us, and we've mapped them onto our PALS atlas surface. And good news is that when we look at areas like their area 44 and 45, they actually line up very well with our manually transferred Brodmann area 44 and 45. But the probabilistic maps are, of course, much more refined and valuable and reliable. We have other kinds of data sets, such as fMRI-derived maps of visuotopic areas, early visual areas mapped by Tutel and Hajikani and many others, V1, V2, V7, and MT+. Another collection of areas reported in parietal cortex by Swisher and collaborators, and you can see an extensive set of cortical areas, fMRI-derived, and you can look at the overlap with when there's the same area reported like V3A, you can see it's in similar but not identical position across studies. So we have issues. It's terrific that we're getting all this data, but we have many maps in partial agreement and partial conflict. Mapping them from individual subjects or summary diagrams to an atlas provides a better substrate for accurate comparison. Now, my only brief foray into monkey land is just to remind you that the issue of areal partitioning has been much more intensively studied in laboratory animals such as the macaque monkey. And here's one version of what started out as a probabilistic map of architectonic areas identified by Jim Lewis in my lab and summarized as a most likely location of areal boundaries over here, where you can view this in our Carrot software and see and identify any of the many component areas by that particular scheme. But the real point in bringing it to your attention is after a century of study in the macaque monkey, we still are faced as a field with a collection of many different partitioning schemes that compete for attention and credibility. This shows a summary of 10 different schemes to show you where there's common ground and where there are a lot of differences yet to be sorted out. Fast forward back to the human cortex. How many cortical areas do we have in your brain and mine? We still don't know. Maybe it's 100, maybe it's 200. We really need to know, but we're faced with many methods, some very powerful, but each with its limitations, different criteria for partitioning the cortex, and many regions that are still pretty poorly charted. And we're faced, of course, with not just the challenge of mapping accurately in any one individual, but the variability in the size of areas and in the folding patterns compounds those difficulties. So what this is to me from a neuroinformatics perspective is motivation to develop a new level of support and infrastructure, what I'm now calling a library of cortical areas. And I'm going to come back to that. I just want to plant that seed and then move on to the even more challenging domain of dealing with fMRI data. So So as a starter, if you just search for PubMed for fMRI, PET, and brain and human, you find something on the order of 20,000 studies, and I've checked, most of them are valid human neuroimaging studies, over 2,000 just last year. So it's not just the sheer number, but also the complexities and differences in how data was acquired, differences in the paradigms, which of course are at the heart of what many studies are all about, but differences in the pre-processing and analyses and localization and visualization. And so, again, planting a seed, what I think we need critically in the field is a set of libraries that give us accurate maps of fMRI data and the critical associated metadata. To give a little more specificity of what I'm heading towards, a typical human neuroimaging study, such as this one by Guy Orban's lab looking at objects versus scrambled objects, you get the data in one subject and see some hotspots of activation, a similar but not identical pattern in subject 2, do it across a population and use statistical parametric mapping to find the significant regions, and then we add the step of mapping these significant activation foci onto our PALS atlas to bring it into a common spatial framework. Well, sort of common, actually. I need to just briefly interject that there are many different variations of a stereotaxic atlas that are used. AFNI, FLIRT, SPM, TAILORAC, the classical TAILORAC, and they differ substantially. This is just a map that we generated to show that the differences in some regions are more than a centimeter from the coordinates for the same basic location. So you can't ignore it, you shouldn't ignore that. And our atlas mapping technique allows us to take data sets, fMRI data, from any of the major stereotaxic spaces that are used, map it onto the PALS atlas, and then compare that with another data set of a different paradigm and compensating for these differences in atlas spaces. And so here are just 3 very different paradigms extracted from the literature. The data has been made available in our database. I'll touch upon that in a bit. But you can see that there are these activations and interesting comparisons start to spring to mind when you can see these side by side rather than pawing through different publications formatted in very different ways. And it's not just behavioral activations, but also the other types of data such as resting state functional connectivity, a cottage industry of itself that can be, that looks for slow correlations in fMRI signals in the resting state and reveals interesting, fascinating spatial patterns of correlations in cerebral cortex. And then just briefly to mention that we've also got the cerebellar cortex into our atlas framework. It's not a population average because it's hard to accurately reconstruct the cerebellum, but we have them tied together in a common framework. And then we are in a position to say, with all that data at hand, how do we get our hands around it and our hands on it? Well, the most common type of data representation is actually not these images, but rather the stereotaxic coordinate, XYZ coordinates that many of you have seen published in long and dry tabulations in a paper, and comparing one table versus another, or comparing visual representations that look very different from one another, makes it tough to do anything systematic in the way of a meta-analysis or just an orderly comparison. But on the good news side, there are thousands, an estimated 6,000 to 10,000 studies that do report these stereotaxic coordinates. And we've undertaken a task where we now want to get community involvement, but we've done the initial seeding of this ourselves, and we have almost 1,000 studies, over 25,000 stereotaxic coordinates, all extracted and brought into our atlas representation. And you can see this confetti-like spray of data from many different studies mapped onto this atlas surface. half of them are actually represent a systematic extraction of all of the neuroimaging studies reporting coordinates in the Journal of Neuroscience. And I'll show you a little bit more about that. So you can see each one is colored according to the study it came from, and the pattern in itself is interestingly non-random. So you can see, if you just quantify the density of foci here, you can see that the hot spots are concentrated in regions like area MT, which is, we can compare it precisely on our atlas map, and also more obscure area IAL in the frontal operculum. What does that mean? It may be experimenter bias and questions asked, or there may be something else interesting. We don't know, but we have a substrate for asking. And we also have a toolkit in our Carrot software for searching this data set by any of the many kinds of metadata it contains. The study-related author and journal keywords, the analysis of what kind of functional task it is. Here's a quick survey of studies related to visual motion processing concentrated in area MT, but showing activations in many other regions. Here's an extraction that goes back to the Williams syndrome folding abnormality. We've got a way to extract just those foci that are centered on the folding abnormality that I showed you previously. And from our limited analysis, or the data set so far, there are 17 studies that report 22 coordinate locations in that region, and you can analyze them as you choose by clicking on a spot and highlighting where it is, clicking on the study and finding study-related metadata, including the abstract extracted from PubMed ID. You can click on the focus and find what table it's located in, what task it was related to, and click on the hyperlink and go back to the study itself and see what kinds of information we don't bother extracting from the study because we make the links to the study. We can also expand this and say, well, for those 17 studies, they had one focus in this olfactory sulcus, but the rest of the foci reported in those studies are actually scattered all over the brain, strikingly broadly distributed. If we do that same experiment, but extract the regions of the intraparietal sulcus as another example, we find many more activations and foci reported, but again, the studies in which those subset of studies are reported are spread all over the map. So we think there's a lot of things that can be done, especially because these data are being made freely available through our SumsDB database. And I will finish up very quickly here. So this database allows you to, SumsDB, you can go to it, and you can, it has many attributes that I'll let you explore for yourself. If you click on the home page here, you can go, you don't get just a list of files, you can actually get pre-constructed scenes that allow you to navigate and see what you're going to be looking at. You can view online without downloading data or software the results, and you can link to journal articles. And we also, in the flip side, have journal articles such as this Williams syndrome study, where the data, there's a link from the online figure to the data in the database, and you can download the data, launch WebCarrot, view the experimental data or additional data that wasn't in the published figure, and you can search for stereotaxic foci. We're going to improve the interface, but the basic capability is there. And you can search for regions of interest in the smaller number of fMRI activations that are in the database. And then lastly, coming to this point about libraries, we think that it will be helpful to the field to organize these data sets and extract from the published literature a subset that are identified as reference areas and stereotaxic foci and various other categories. And we are midway through this process, but have a lot we want to do. And we also want to tie in our own database efforts with the emerging neuroscience information framework that was pioneered by Dan Gardner and others for a couple of years, and now Marianne Martone will be leading the charge for the next phase of development of this broader neuroscience access, this portal for accessing a tremendous variety of neuroscience resources, and bringing us to an age where there's a huge amount of data analysis and data mining that can be done with tools at hand or soon to be developed in the coming years. So thank you very much for your attention.